Thank you for the introduction. Thank you for stopping your lunch sooner than optimal, but okay. So I want to talk to you today about some ideas that. Okay, some ideas related to one of actually the grand challenge that attracted me to biology from the beginning. So it is this challenge of computing gene regulation. This maybe naive ambition to take the information that is buried into the genomic sequence and somehow being able to compute biology. So for me, this was the idea of computational biology when I started it about 14 years ago, something like that. So it turned out to be, of course, very naive, and it's a very difficult task. And I want to try and identify with you some of the keys for why is this so difficult, why is this so naive. So I think a first important factor is that genomes are physical elements. They are molecules. They are not just combinatorial objects. They are not just information. Well, a second major factor is that cells are not acting based only on this information or the DNA sequence, but they in fact act based on a lot of other information or memory that they acquire in the process of development or other processes in biology. And the third effect is that gene regulation is not really a combinatorial process. It's actually a highly stochastic process that involves small numbers interaction between different types and of molecules within the cell. So we cannot really try to think of it as a deterministic function that take information and generate something. And the fourth reason that we will not be able to do too much with is that the 100 trillion cells within the body of each and every one of us have more compute power than my cluster at the Weizmann or the cluster any of you have, okay? So we will not be able to solve this. We'll have to deal with it somehow. But I want to try and go with you today about several approaches we are developing in the lab and others are developing around the world in trying to pinpoint some of the difficulties here and devise not solutions, but strategies to start and deal with them. So we will discuss massively parallel single-cell RNA-seq as a way to deal with the stochasticity and the heterogeneity of the gene regulatory program. We are going to discuss single-molecule DNA methylation models in order to try and tackle the fact that cells have memories and we cannot decipher the program based only on the sequence. And we are going to discuss chromosomal conformations and models that are using single-cell or single-molecule high-resolution IC to try and tackle the fact that this information, the genomic information, is buried into some physical form. So this is the plan for today. Let's start with massively parallel single-cell RNA-seq, which for me, it's a hugely exciting field that is booming out there in the last 2 years or something like that. So the starting point here is the fact that cell heterogeneity or the fact that cells comes in different types is something we all know of when we read our first biology textbook. But the definitions or the phenomenology of these cell type phenomena is not really well defined or is based on some discovery process that is inherently limited. So usually the paradigm is to identify some kind of marker. It can be something that you define under the microscope or some molecular marker if you want to use FACS. Then you are selecting your marker, sorting according to the marker, and do some functional analysis on sorted subpopulations. And this is, of course, highly prone to circular logic because you need to define the markers in order to have the cell types, but then you define the function of the cell types only based on the markers. Of course, you can do iterative cycles in order to refine this. So you identify marker X, you sort by marker X, You go and identify some functional program in a subpopulation. Maybe you identify new markers. You go back. And indeed, we know a lot about biology using these techniques, but somehow it seems to be limited, especially when things become more and more sophisticated. So the single-cell RNA-seq paradigm allows you a very, very different approach to this classical problem. So what you see here, let's start with that, are about 2,000 single-cell RNA-seq profiles that were acquired using mouse spleen cells that were enriched for dendritic cells. And you see here the covariance or the correlation between those single-cell RNA-seq profiles. And the idea in such an approach, once you have the technology, is that you can sample cells in an unbiased fashion without assuming anything or minimally assuming something about their behavior, and then assemble them into groups or define the cell state space in an unbiased fashion so you can discover cell types in a very native and natural way. And of course, you can also discover internal structure within the cell type structures. So what you indeed see here are beautiful clusters within this splenic population that represent cell states that are highly correlated to each other. And this is published work, but if you follow up and do targeted FACS sorting for known markers, you see that indeed many of those cell types or groups of cells indeed correspond to very well-known cell types like B cells or NK cells or monocytes. So in fact, what we did here in the project that was done in a collaboration with Ido Amit at the Weizmann was to tackle a very, very easy problem because we took a bunch of cells that are highly distinct from each other. So there is not a real problem to identify or to distinguish a B cell from a dendritic cell. Anyone can do that using a variety of methods. These are very different beasts. But within this large cloud of cell states that, for example, represent dendritic cells, there are a lot of additional variabilities, covariances, and things that define the dendritic cell state. And this is where single-cell RNA-seq is going to become particularly powerful. So before going into some updates and new data that is coming out of this approach, I want to go over with you on how we want to model those relatively complex experiments. And I want to make the point that here, as in many other modern experimental technologies, we have the advantage of knowing and relatively well controlling many aspects of the experiments, and we can aspire, unlike before, to have a relatively complete understanding or modeling of the different stages, both in the experiment and in the biology that generated it. So let's go over the single-cell RNA-seq pipeline together. So we are starting with a cell population. This cell population, to begin with, is the biology, okay? But we are not treating this entire cell population, unfortunately. We will always sample a subset of it. So here, immediately, we are adding some experimental design and some biases that may change the experiment. The next phase is we take those sample cells. In each of them, there is a limited and relatively small pool of RNA molecules, about 100,000 or 200,000, not more than that, poly-A tagged RNA molecules. We cannot sequence all of them, okay? So we have an additional level of sampling. Oh, the Mac to Windows, never mind. So we have an additional level of sampling that take this limited pool of RNA molecules within the cell and give us only a fraction of it. Later on, we are amplifying them massively. And after amplifying them massively, we are pulling together usually many products of many different cells. So again, an additional experimental stage that can introduce biases. And by the end of the day, we are sequencing this pool, okay? Again, applying some technical decisions as to what depth we are going to try and sequence. So ideally, we want a model that capture both the biological factor and the technical factors that generate data from such experiments. So the biological factors and the biological variabilities here are, of course, the cell compositions that we are looking at may be variable. So maybe in one experiment we have more B cells and in other we have more NK cells because the mouse is different, because the mouse is sick, because I don't know why. So these are real biological factors. Then we have other biological effects that have to do with the actual cell type and cell state within the population we sampled. For example, if the mouse is sick, maybe B cells are looking a bit different at this particular experiment. Again, the nice thing about single-cell RNA-seq is that at least the cell composition is something that we will be able to control or identify when we have the data from the experiment, something that was never available to us in standard microarray or gene expression experiment where we only had the ensemble average. Of course, there are many, many technical effects that change the outcome of an RNA-seq protocol. These have to do with the enrichment, dissection of how you separate the cells, how much time you waited to the fax, all kinds of lysis, delays and degradation, you name it. Many, many different effects can change the way your experiment will look like. And most importantly, or very importantly, by the end of the day, you are not fully sequencing all the molecule within your cells, you are sampling. You are always sampling. So for us, a reasonable model that describes this experiment is starting from the end, we are doing some multinomial sample of a finite set of molecules. This multinomial sample is coming from a distribution that depends on the cell we are looking at. So if we have cells for many different types, each of them will span for us a different multinomial distribution. And much of the interest in single-cell RNA-seq, thinking of it as computational biologists, comes from the distribution of those multinomial distributions. So biology defines for us some distribution of molecule pools that are being determined by the actual biology, and this is what is of interest for us. So a big open problem in this field, which is young and emerging, and you are all encouraged to join in, is to try and find good models that will describe for us how these distributions behave. For example, come up with interesting network models that will span up a space of multinomial distributions that make sense for biology and plug them into this experimental model and take into account both the sampling variances and the biology variances in order to make sense of such experiments. So quickly, some example on how such data can look like. So in the previous examples I show you of the dendritic cell or the splenic mouse cell population, you can stimulate the mouse with LPS that imitate infection, and you see here you see in different clusters or different cell types that were inferred from the wild type untreated mice, you see the response of many different genes, okay? And you see that some clusters of genes are very specific in the response to a specific cell types. So if you only had this upper part of this diagram, it would mean that the cell types were completely preserved. On the other hand, there is a very large group of genes that respond laterally. In many different types, they are responding, or different transcriptional modules are being activated in many different types. So for example, if you would have come to try and identify cell types in LPS-treated mice, you may have come to different conclusions than what we originally came to, because all those effects may have caused you to think that these are a cell type, but they are not. They are just modules that are being activated in many different types. So this immediately goes to show that the type concept is something we need to be very careful about. So there are very interesting genes here, of course, but they are representing very specific modules that can be activated in many different cell types. A similar phenomenon only from the other direction is that cells from the same type, for example, dendritic cells, once they are activated, can be dissected or be diversified into many different types of responses. Here you see dendritic cells in this mice splenic population after LPS treatment, and you see that there is a group of cells here, 500 cells sampled altogether. Some cells are responding based on TGF-beta signaling, some of them based on TNF, others are invoking the general interferon response. So all of those cells came, as far as we understand it, from the same type, but they are responding very differently. So these are of course not different types, right? So again, we need to examine carefully the way by which we define the cell state space. Here is a much more complicated example. This is new data coming from an attempt to understand hematopoietic development in the mouse system. So hematopoietic development is relatively well characterized, or so at least we think. It is being characterized by this wonderful tree that represent a set of developmental decisions that are by and large thought to be hierarchical. So you start with the stem cell, then you differentiate to either a myeloid or lymphoidic fate. Within the myeloid fate, you can go to erythroidic fate or to different granulocyte and other cell types. Okay, this is the classical Weissman model for hematopoietic differentiation, one of the most beautiful stories we have out there in biology. But now we can assess this model critically and ask ourselves, is this really what's going on, okay? So we sorted witidoamide people, many cells that came from these regions, CMP, MEP, and GMP, and we just performed massively single-cell RNA-seq on them. Here you see some 4,000 different cells. And we asked, are they going to reconstruct for us this beautiful hierarchical structure? So we discovered that partly yes, partly no, okay? What you see here are is the actual gene expression, many genes and many different cells. Here you see, I hope you can see the colors well, you see the correlation matrix just to visualize the basic groups we have here. So this is clustered based on some EM-based algorithm that try to learn those mixture of multinomial that I alluded to before. The details are not very important. The algorithmic details may not be very important. What is important is that we have pulled together here cells that were pre-sorted as erythroid progenitors here, or myeloid progenitors here, granulocyte myeloid progenitors, or a group, or larger group of cells that were supposed to be the common myeloid progenitors. And we clustered them together. So indeed, the MEP and GMPs formed very distinct clusters. But now when we looked at the CMPs, we see different and very interesting behaviors. First of all, we see a large group of CMPs that resembled by and large GMPs. So they seem to be toward the fate of going toward GMPs. They are characterized by some transcription factors like CBP, alpha, and PU1. So the nice thing about single-cell RNA-seq is that once we have data like that, we can immediately go and try to see what kind of regulatory model generated it. And we do not have to rely on markers at all, okay? So indeed, CBP, alpha, or PU1 are factors that seems to drive the GMP fate. So that makes sense. Here is the complementary set of cells that seem to resemble more MEPs than GMPs. So these are CMPs that seem to go toward the MEP fate. They have GATA2 as active, not so much GATA1, and a weak KLF1. GATA1 and KLF1 are the prime transcription factor that control the MEP fate. So we do have some, it makes sense. The Weissman model at least partly makes sense here. But we also see all kinds of other strange groups of cells there. Some of them seem to be of a megakaryocytic progenitor type. And some of them seem to be like dendritic cell precursors. So things are much more complicated than a simple hierarchy that need to decide between fate A and fate B. So it is going to be very, very complicated and very, very interesting to understand the gene networks within this complex cell population. But I think we already understand that instead of this hierarchical model of cell fate and cell differentiation, we are going to have to come up with models that are much more fuzzy and diffused. Instead of going from fate A to fate B to fate C, I think that it would make much more sense to think of clouds of states, for example, the progenitor fate that can travel between different variants of the progenitor state, and occasionally, maybe when reaching to the right niche in the bone marrow, get the signal and jump directly to one of the more differentially terminally differentiated fates, not necessarily going through different hierarchies of progenitor states. It will remain to be seen, but it's going to be very, very interesting. And the technology is here to actually ask this question, which is, at least for me, very, very exciting. Okay, so we discussed how to try and deal with stochasticity in the gene expression or the regulatory program, the genetic regulatory program. But we also said that it's hard for, so this gives us the ideal data points from which we can try and dare and go and infer regulatory model. Because here, for the first time, it wasn't like that before. We actually have snapshot of how this regulatory program is working, okay? Previously, we had to settle for averages over millions of such states, which were a priori mixed between, as you see here, mixed between very, very, very different conditions. So at least there is some hope here. But as we said before, it probably is not going to be enough because we cannot think that the sequence alone determine the program. We need to somehow map also the memory. So DNA methylation is a very important part of this epigenetic memory mechanism, so we think. And the literature on it is, in many, many cases, very, very confusing. So if you think of development starting from the embryo, going to early differentiation and going to terminal differentiation, people usually in the textbook write or idealize the situation as the embryo being reprogrammed to a certain original epigenetic state. And then through a series of differentiation that are being driven by transcription factor or other and epigenetic factors, DNA methylation is hypothesized to be gradually changing to reflect commitment and differentiation along those lineages here. And it's, for us in the group, for a very long time, it's very interesting to try and critically assess this and see what is really going on here. Is it some deterministic process taking state A to state B to state C? Or maybe something which is more complicated and stochastic? So for that, we have developed a set of methods, both computational and experimental. One very important improvement we have introduced into the routine acquisition of DNA methylation profile is the addition of molecular identifiers, unique molecular identifiers, UMI-bis, that allow us to take standard bisulfite sequence, which is the technique by which people are mapping DNA methylation profile, and get rid of all the terrible PCR artifacts that we always had there. So instead of being able to just compute average methylation for a certain CpG or CpG alanine in a genome, we are now able to assess with a lot of confidence and known statistical properties the frequency of each methylation pattern in this locus. And this is something which for us is very important, because what we want to try and distinguish are situations where a certain locus in the genome have a perfect, coherent DNA methylation profile. So all of the patterns are just one. Compare it to what we call the unimodal locus, where there is one dominant pattern, but then some noise around it, a noise which behave in, let's say, in independent fashion. So the probability of each pattern then will be just the multiplication of probabilities of noise in each of the loci each of the CpGs in this loci. We want to compare it with other potential models of DNA methylation, for example, the bimodal model, where two patterns are dominating the locus, and maybe there is some additional noise around each of them. So here it will be some kind of a mixture model around two centers, and maybe more complicated scenarios, of course, that you can generalize from this. Why are we interested in, for example, bimodal patterns? So one type of bimodal pattern can simply represent a monoallelic situation, like you usually see in imprinting. So this is interesting, but not our focus here. On the other hand, if you can prove that there are certain places with bimodal behavior and a mixture coefficient that is definitely not 1/2, it means that there is probably a subpopulation there that behave differently, and there is some interesting thing going on with the determination of memory within this population. So this is one important goal we have with UMI bisulfite sequencing. So using these techniques, we went to try and see to what extent this model of single epigenetic state in the embryo differentiating into, in a deterministic fashion, into many different epigenetic states in terminally differentiated cells, we wanted to test if this model makes sense or is holding. So we did our take on the classical mutation accumulation approach that is common in evolution. We did an Epi-mutation accumulation approach. We take single cells, we expand them to clonal population, and we use UMI-bis bisulfite sequencing to test the variability or the lack of coherence or lack of coherence in these clonal populations. And we first applied to embryonic stem cells, which are one possible model to the embryonic state. It's a problematic model, but to begin with, let's use it. And we tested the frequency of our methylation patterns in the original polyclonal population to the frequency of the same pattern in a population that went through a clonal bottleneck. And we found no difference whatsoever. So this allows us to determine that there is no significant transmission of epigenetic memory at this cell state, at the stem cell state and probably at the embryo. On the other hand, and very interestingly, there are different loci in the genome that maintain very different frequencies of the dominant methylation pattern, which suggests to us, if you think of it as a probabilistic turnover process, that the turnover process, the kinetics of the turnover process is generating something which is almost deterministic. So there are going to be regions in the genome that have a certain level of coherence, which is very high, okay, almost always the same. And there are going to be regions in the genome that are always very noisy. Why? Because the balance between the process that put methylation there and the process that take it away is different here than it is here. So it's all a matter of the stationary distribution to which the process can converge to. So this brings me to try and hypothesize that instead of a single deterministic epigenetic profile that happens in the embryo and later differentiate, we see something which is different. We see a rapid turnover process in the embryo that is happening by balancing addition of methylation on non-protected site and removal of them, and we now know the enzymes that are actually doing this. But this balanced process is now changing once the process that removes methylation is being shut down relatively early in development. While this is being shut down, the process that writes methylation is still active. And at this window of opportunity, the embryo already starts to make developmental decisions. So to generate different lineages, to occupy, for example, different enhancers. Once this is happening, what you get is a negative image of the regulatory state at those early differentiation or early differentiation decisions. Simply because the process that remove methylation is shut down and the process that edit is still active. This is very beautiful and allow the embryo to maintain its early decisions without any instructive mechanism that recruit methylation there in a targeted way. Okay, we can now continue and ask what is happening later. How is this memory evolving later? I will be relatively brief here. Papers are published. What we see is that in the somatic cells we looked at, there is clonal transmission. So here, if you are taking the polyclonal frequency of a pattern, you'll find patterns that are rare in the polyclonal population, but if you are going through a clonal bottleneck, they become almost half of the population, meaning that the clonal population is maintaining the methylation pattern of the founding single cell clone. You can use such data, I'm not going into the detail, to actually infer some like semi-coalescent model to the exponentially growing clonal population, and from that infer turnover rate of how quickly this population is putting in or erasing DNA methylation profiles. And this generates very interesting conclusion. For example, you see that the rate of losing methylation, please ignore this part of the slide for now, the rate of losing methylation very, very strongly depending on the timing of replication of this particular region in the genome, okay? So regions in the genome that are very early replicating in the right here, are losing methylation much, much less quickly than regions that are replicating late. And in the long run, so these are relatively slow processes, so we are losing a methylation every 200 or 300 cell divisions. But if you think of an aged cell, okay, the effect can be profound. And we think that the majority of the report, for example, on changes in methylation in cancer or in aging are a consequence of such slow turnover processes. So to put this into the model, in addition to this embryonic freeze where the addition of methylation is continuing after the removal of methylation stops, at a certain state, both of them stops, methylation become relatively fixed, and then only a slow process of losing and gaining methylation just due to noise is continuing to modify the methylation landscape. Okay, so just to put another piece of observation on the same idea, you can, for example, take population of cells like B cells or embryonic stem cells and do very, very deep bisulfite sequencing on them with unique molecular identifiers and try to find regions that are bimodal or not bimodal. And in most terminally differentiated populations like B cells, almost all of those bimodal populations would actually represent monoallelic states. On the other hand, in the embryo that only recently went through this freeze, you'll discover many, many different bimodal populations that are not at the 1/2 mixture coefficient that represent monoallelicness. I think I'll skip this part in the interest of time, which have to do with an addition to the model that tells you that in fact, methylation is not completely stable after you finish this embryonic differentiation, but there are additional changes that happen exclusively at enhancers that can lose their methylation if they are becoming activated. And the crucial observation here is that when those enhancers are losing their methylation, even if it is happening at some progenitor state, this methylation would be propagated, or this lack of methylation would be propagated throughout the continuation of these lineages. And in none of the cases we looked at so far, we haven't discovered significant, and I'm not talking about cancer, significant gain of methylation in a targeted way. But we do discover loss of methylation or hypomethylation in most of the enhancers family we looked at. Okay, so finally, I want to discuss the physical aspect of the genome. So we discussed the fact that cell states are stochastic, and we need to look at individual cell states if we want to try and model regulation. And then we discussed the fact that cells carry some memory, so we want to understand how this memory is behaving, propagating, being determined. I haven't touched the question of whether, for example, methylation is actually affecting regulation. It's a difficult question. But at least we have some understanding on how the dynamics is looking like. Now we want to touch the point where DNA sequence is not enough because it is being interpreted within the physical domain of the nucleus. So an important discovery in that respect that was made 2-3 years ago by us and others showed that instead of linear chromosomes in which all elements are equal, chromosomes are divided or demarcated into something that was nicknamed topological domains. You see those squares here. What you see here is a high C matrix that represent the intensity of contacts between different chromosomal elements. Here it's just 2 megabase from the Drosophila melanogaster genome, but essentially you see it in all animal genomes we looked at. And those beautiful domains are very, very intriguing in the sense that they can serve as regulatory units. Okay, they can serve as regulatory units in which genes have their own mini genome. And now instead of having to deal with 200 megabases of sequence in chromosome 1 instead of that, and then deal with all the misspecificity and the spurious binding site and all the stuff you may have there, a gene can mostly worry about the stuff that is within its topological domain. So now we can go back to a classical question, which is the question of enhancer sequence specificity. So we heard today talks, and there is a lot of work in trying to understand how the local sequence content of an enhancer will affect its activity. But we also know that the place of this enhancer or sequence in the genome may be very important in order to understand what is going to happen with this enhancer in a particular cell type. So here's one piece of evidence from a recent work we've done on flies, going to a classical paradigm of epigenetic specificity, which is Polycomb domain recruitment in Drosophila. So the details of this particular system are not very important, but just take those following facts. There are two sequence-specific factors, DSP and FO, that can identify their own motifs in flies. These are supposed to identify sequence elements that are called PREs. And the idea is that PREs then are nucleation sites for the folding of three-dimensional Polycomb domains by forming contacts between themselves. The problem is that the sequence specificity of FO and DSP is not enough, okay, in order to predict where Polycomb domains would occur, even in the small, tiny, and very convenient fly genome. It's just not enough. You cannot, you get super statistically significant signal, but the genome is not settling for statistical significance. The genome is working at 100% specificity and sensitivity. So something is working there, but we cannot understand it. So what is happening there? Our hypothesis now is that what is happening is that those PREs, what you see here, are average high C contact between enhancers that are marked as PREs, comparing to other types of element, and you see that those guys tend to contact each other. So far we were always thinking about this contact as something that comes after the fact, okay? You recruit the PRE, and because you recruit the PRE, you form the contact. Now the new idea is that this can also be reversed. Think of a hub of chromatin that see many different sequences around it. Now the question of sequence specificity becomes very different. If you have some level of cooperativity there, even if the specificity to local elements is relatively weak, the common specificity of the hub can be very strong. So it seems that the folding and specificity questions may be strongly linked with each other. So something like that, weak sites out of the Polycomb domain context will not form anything, but weak site within a Polycomb domain, because they are capable of forming three-dimensional contacts, would cooperatively generate something which is very strong. Of course, this is not much more than a hypothesis at the moment, and we really want to test it and to develop methods that will allow us to critically assess the links between three-dimensional organization of enhancer hubs, promoter hubs, and their specificity. And in the group, we've recently started to come up with a possible solution for that. So this is very new. I'm very excited about it. I'll just show you a bit of it, okay? Here is a high C map for human. It's for K562 cells. This is the diagonal, so this is like the chromosome. This triangle you see here is a topological domain. It means that this piece of the chromosome is highly contacting with each other. By the way, due to some improvements in high-C technology, our resolution now is much better than before. And for example, you can see this point here that represents preferential contact between two elements at the border of the hub. These are CTCF sites that close the loop of the topological domain. So we have here a couple of genes, and these are the CTCF, the strongest CTCF sites in the region. And this is always what we had so far, based, if you want to really know, based on accumulation of some 500 or sometimes 1 billion or even 2 or 3 billion individual pairwise contact events. So these are individual nuclei in which the two genomic elements happen to be in the same place in the same time and therefore got linked together. This is what high-C is trying to quantify. What we can do now is instead of, or in addition to that, generate not pairwise interactions, but groups of many different interactions. So we call these contactamers. So these are in fact 3C complexes that were always in fact there in the 3C protocol, only that we were never been able to assess them properly. So in addition to this beautiful high-C graph, you also have a chain of elements, like a walk along the chromosome. Those of you that did genome assembly or something like that may be familiar with such graphs, okay? So you can try and reconstruct now not only the pairwise or the statistics of pairwise interaction, but in fact, the precise way by which this particular, or one nucleus, had its conformation for many, many different pieces of chromosomes. So I think for me, this is super, super cool. You see here different contacts that are lining up within the topological domain. So just to show you that this can bring up some very intriguing results. Here, for example, an example of a concatemer where we have 4 different GATA sites, 4 different GATA enhancers that are sitting absolutely together, okay? We're cross-linked within the same 3C contactamer. We have a lot of such examples, but of course, it's very, very challenging to try and analyze them, and I won't make any generalization here today due to lack of time, but just think of it. I think it's going to be a very beautiful problem to work on, somewhere in the middle between the statistics of pairwise map and full-blown like chromosome folding models that are that people are also developing. So to conclude, of course, we will not be able to compute gene regulation, okay? I'm not trying to say we will, okay? But we can make some progress toward it by developing techniques that will attack what I think are the most crucial problems in that respect, which are identifying the actual state and generating a large number of measurements of single-cell transcriptional states, understand memory mechanism properly and their dynamics. Of course, we have to study very carefully the relationship between the two, and treat the genome not as a combinatorial object, but rather as a physical object. So with that, I'll thank the many people in the group that contributed here, Zohar Mukamel and Zohar Shiponi for work on methylation, Effie Kenningsberg and Yara Arking for work on single-cell RNA-seq, Yaniv Lubling and Pedro Olivares for work on the high-C, and our collaborator, Ido Amit, first and foremost, at the Weizmann, and Giacomo Cavalli for some of the fly results you saw. And you for your time. Thank you.
Quite nice talk. I just have a quite simple questions about the single RNA-seq you just proposed. It seems that, because to my knowledge, it seems that for single RNA-seq, we lost a lot of information, maybe even more than 50% of the information due to randomly. So how do you tell whether the difference in different single cells is truly the difference in the cell itself, instead of just some technical noises due to the random loss of the signals?
So if I understand your question, you are referring to the fact that we are not sampling all the RNA molecules within the cell. So of course, we are not sampling anywhere near all the molecules, and there are people that are promising to sample like 50%. I'm saying that we are sampling 2 to 3 to 4%, okay, not more than that. But the crux of that is that, in fact, there are millions of cells out there, okay? And even if you're picking up only 5% of the molecules, you still have couple thousands of measurements, and the correlation structure that you are recovering allow you to reconstruct the distribution of molecule counts within particular cell types with accuracy which will be as high as the number of cells you have, okay? So we have to understand that even the real biology is not giving us some deterministic number of molecules per T cells. It's simply not like that. There are variances all along the way, okay? So I don't see it as the major problem, and in fact, in terms of information contact, I really prefer protocols and approaches that collect more cells than protocol that make a bigger effort and collect more molecules per cell. For most of the applications we are now developing, this is more important.
Thanks.
So I have another question about the single-cell RNA-seq. It was in the hematopoietic system, and presumably you mentioned that the single-cell RNA-seq lined up well with the FACS results. So FACS is also a single cell, and I'm curious if the results of the FACS were tracked with the RNA-seq, and if these kind of the common progenitors, if their surface expression of these surface markers correlated with these kind of undefined populations at all.
Yeah, that's an excellent question. You know, the fact that we are using FACS for many years to characterize those populations was because this was the only thing that we could do, okay? But many of the markers that are being used, for example, to define progenitors, are not the direct functional effectors or the molecules that we actually think that are driving the progenitor program, okay? They are just markers. So by and large, there are excellent correspondences between, you can do index sorting and you can really see that what you are identifying the single-cell RNA-seq is perfectly consistent with the markers you do have. But the problem is that there is no ontogeny of the markers. So the markers are not sitting perfectly beautifully on the tree, okay? You can have a bunch of cells that express a marker that was considered as a marker for progenitors, but they are actually already thermally differentiated, and you will not have any way of identifying it from the FACS.
I mean, that's the question, though. You have the single-cell FACS data, and you have the single-cell RNA-seq data, so if you have some lineage, some marker reuse...
Precisely the examples you saw are examples where, for example, the megakaryocytic progenitors there are most likely coming out of place, okay? They are probably more differentiated and they simply have the same surface marker. It's not an error of the FACS. They have the surface markers, but they are not the progenitors.
Thank you.
So you show all these interesting results from single-cell RNA-seq, and then you also showed, look, the single-molecule methylation, found these interesting patterns over development. And I was wondering if you've thought about doing single-cell RNA-seq over development, looking at different populations and seeing if you can start to understand some of the effects of the different methylation patterns you're seeing.
Of course, we are doing these experiments, and they are very interesting. But I haven't went into my individual thoughts on how methylation is exactly functional or not, okay? Mm-hmm. It's a very tricky business. If I just have to give one answer, then as a first approximation, methylation is not functional, okay? Of course, I think it is doing something, but it is not silencing promoters and it is not silencing enhancers. It is, currently we are mostly using it as a marker. A very interesting marker, but a marker.
Thanks.
Amos, thank you for a beautiful talk. I'm curious about the tree representation and how much that can be perhaps rejected by your data. So if you look at individual cells, you can actually start asking if they're consistent or not. Like if you start putting them all together into some kind of tree, you can basically start asking, is there a potential tree that would explain this? Or can I actually now reject the concept of a tree and think more of a random walk? So are you at that stage where you could actually make the statement that no, a tree is actually not an appropriate representation?
Theoretically, yes, we are at the state in terms of data, okay? But this is much too fresh to be able to reject such profound hypothesis at this stage. We have to do a lot of more work in order to say this confidently. I invite all of you to think theoretically if a tree concept is at all makes sense and is consistent with what we know about biology. Okay? One thing we are completely ignoring with all those techniques is, for example, the niche. Okay? The place where the cell is, the signals it is getting from the environment. This is something that single-cell RNA-seq is not solving for us. Okay? And if you are thinking about the niche as a major effect or a major determinant of those developmental transitions, I think that you will have to let go with the tree concept. But I'm willing to continue and discuss this.
This is just a comment for this question and also this one over here. The fact is I think your data are in magnificent agreement with a lot of the data from many labs in the field, and so it is, the CMP has always been controversial, and so I think many people are, because those markers in fact are not a well-defined population even in flow cytometry. They're a bridge between populations, so I think many people are very happy with your results.
Yeah.
right? Distinguished species